This video is going to talk about the structural stain called the capsule stain. Back in Unit 1, we talked about the accessory structure known as the capsule, which is this sugary layer that a lot of pathogens tend to have because it helps them evade phagocytosis, which is part of our immune response. So it's very advantageous for a bacteria to have a capsule. So we, scientists that is, decided to develop a stain to determine whether or not an unknown organism has a capsule. So the capsule, as I mentioned a second ago, is this sugary layer, right? That's tightly associated with the organism. But the overall charge of the, charge isn't the right word, but the capsule overall has a neutral charge. So it's also made up of molecules that typically repel a lot of their stains because of that neutral charge, right? Those positively charged simple stains aren't going to be attracted to the capsule, nor is the negative stain. So scientists had to get a bit creative to figure out how can we view this neutral structure that's made up of sugar. Well, the capsule stain actually uses both an acidic stain and a basic stain. So essentially, what's going to happen is they use an acidic stain, which is one of the two types of negative stains. So either Congo red, which is red, like you see in the picture, or nigrosin, which would be black. And that is going to stain the background, just like we talked about in the negative stain. But then they also use a basic stain, which is going to have a positive charge and be attracted to the actual cell. So what happens is the background gets stained one color, the actual cell gets stained another color, and the capsules remain unstained. So they appear colorless and they look like little halos when you're viewing them under the microscope. So for instance, like these little colorless areas right here, these are the capsules. Okay, so they say they look like halos. So with the capsule stain, you cannot heat fix these cells. Okay, so you don't do heat fixing as part of the smear process because it causes the cells to shrink, but it can also cause this artificial halo. But think about what happens if you heat sugar, right? It's actually going to dissolve the capsule. Okay, so that's why this artificial halo is going to give you a false positive result. So you do not heat fix if you're going to do the capsule stain. All right, so what do you do? Well, if you kind of follow along with this slide, okay, so you're going to go ahead and put some Congo red on the right-hand side of a slide. And then you're going to use aseptic technique to mix in some of your cells with that Congo red, okay? And then you're going to take a second slide and you are going to slide it along the surface, okay? And to form this nice 30-degree angle. And then you are going to kind of like draw it back into that drop of red on the right-hand side of the slide, okay? And at that 30-degree angle, you're essentially moving the slide back. Oh, my pen's not working. You're going to move it back this way, and you're going to be drawing out some of, or spreading out some of these cells that are mixed with that Congo red. And so you're trying to get a nice gradient, right? So that with the microscope, you can view it easily under the microscope. Okay, so you use that second slide to spread the cells in the dye across that bottom slide, okay, you're going to dispose of this one in the container of bleach that's on the lab tables because it has touched the cells that were mixed in with that Congo red. And now you're going to let this air dry. Okay, again, you're not heat fixing because that will dissolve the capsule and cause the cells to shrink and potentially give you an artificial halo. Once it has completely air dried, and that's another reason that you spread these cells out, is so that it can actually air dry in a timely fashion. Okay, so then you're going, once it's completely air dried, you're going to place it on a staining rack, and you are going to cover it with a positively charged stain. Manival stain is an option there, and you let it sit for 1 minute. And then notice what you're going to do here. You're going to dip the slide into a jar of distilled water a couple of times to get that stain off. Okay, and then you are going to tap the edge of the slide on a paper towel to remove that excess water, and again set it down to let it air dry. Because you did not heat fix your slide, those cells are not going to be attached as firmly to the slide. So you don't want to spray it with water. You don't want to actually blot it dry either. So there's a lot of sit and wait for this stain because you have to let it air dry. But if you look closely at step 9 here, you see how it's really like a darker blue on the left side of the slide and it's lighter on the right-hand side of the slide. Well, this is that region that you're going to want to look at under the microscope because that's where the cells aren't going to be quite as thick. Okay, but if you looked there and you didn't see any cells, then you could scooch farther down to the left, but it tends to get really dark as you look more to the left. All right, y'all, that's the end of this video. Let me know if you have any questions about the capsule stain.